Hello, this is this lecture on hemoglobin, myoglobin, heme, and oxygen transport. The main topics we're going to deal with is the interaction of hemoglobin with oxygen and what properties of hemoglobin allow it to serve as a very good transporter of oxygen, one that binds it in the lungs and then releases it in the tissues. We'll also touch upon myoglobin, which is a related molecule but has distinct or different functions, although similar but different. Heme is a major constituent of hemoglobin. We'll talk about its synthesis and catabolism and the relevance of that to certain disease states. Now, as you know, the major function of the red blood cell is to transport hemoglobin and therefore oxygen, because hemoglobin is its carrier, from the lungs to the tissue to provide oxygen for the functioning of our tissues. Myoglobin, on the other hand, is found in muscles mainly, the cardiac muscles and skeletal muscles, and it also binds oxygen. But different from hemoglobin, it doesn't release it as easily. So myoglobin serves more like a storage form for oxygen that can release it when there is extreme need of oxygen under conditions of oxygen deprivation for the tissues. We should also mention here that hemoglobin is not only an oxygen carrier, it also, as part of its function, serves as an acid-base buffer. And in fact, the RBCs are responsible for most of the buffering effect, acid-base buffering effect of the red blood of the blood. Now, this is hemoglobin. Hemoglobin, depicted here, as you can see, is made up of 4 globin monomer chains. 2 are in blue and 2 are in purple. Each of these contains a heme group. So there are 4 heme groups in every molecule of hemoglobin. In contrast, myoglobin consists of only one chain, one globin chain with one heme group. So that's the first difference. As you can see, the chains, the globin chains, are named here alpha and beta. So 2 betas and 2 alphas are what form the normal hemoglobin that is found in human beings, which is called hemoglobin A. There are other chains that can be formed, and they can lead to different kinds of hemoglobin, and we will mention these later on. And the heme molecule that you see in the middle here, the red dot with a yellow surrounding it, is actually depicted on the left in the blue circle. It consists of a porphyrin to which iron is bound in the middle. A porphyrin molecule, it's called protoporphyrin 9, linked to an iron atom. That's heme. So how is hemoglobin synthesized? The first step is the synthesis of a pyrrole ring or pyrrole molecule by the combination of 2 succinyl-CoA and plus 2 glycine molecules. Succinyl-CoA coming from the Krebs cycle. Each pyrrole thus formed will bind, four pyrroles will bind together to form a protoporphyrin molecule, protoporphyrin 9, which then binds the iron to form heme. We'll talk in more detail about this process. And then heme combines with the polypeptide to form the chain, the hemoglobin chain. And then 4 of these chains bind together to form hemoglobin. And there, as I said, there are different chains. In human beings, alpha and beta are the main units. So heme is a non-protein prosthetic group, and it's found in many proteins, not only in hemoglobin. It is found in hemoglobin, it's found in myoglobin, but a number of other enzymes like oxidases, catalases, cytochrome enzymes contain heme and essential for their function. Myoglobin and each of the hemoglobin chains bind to heme in a non-covalent manner. And you probably know that heme is the reason why we have red as the color of blood, and meat, which is myoglobin in the meat, in the muscles. So that's where oxygen actually binds to the hemoglobin. It binds to iron, which is in the heme molecule. This is a more detailed view of heme synthesis, which is also called porphyrin synthesis, because most of the components are what are called porphyrins. They have a structure, a specific structure. So the first step, as we said, takes place in the mitochondria, and it is the formation of delta aminolevulinic acid from succinyl-CoA and glycine, which is metabolized by an enzyme called ALA synthetase. This enzyme is a rate-limiting step for the formation of heme, and it is regulated by the levels of both iron and heme. In other words, when there is a decrease in heme levels, there is a stimulation of ALA synthase to form more heme, and vice versa. Now, once ALA is formed, aminolevulinic acid, it moves out into the cytosol, and several steps take place there. We're not going to go into detail about the enzymes and the intermediates until you reach to coproporphyrinogen 3, which then goes back into the mitochondria to form protoporphyrinogen and finally protoporphyrin 9, which is the compound that chelates iron through the enzyme ferrochelatase to form heme. So heme thus formed in the mitochondria then moves out back into the cytosol, where it links with globin chains that are being synthesized by the regular peptide synthesis machinery of the cell. And there are multiple globin chains coming from multiple genes, as you know. This combination results in the formation of hemoglobin, and hemoglobin then gets incorporated into the This is a more detailed slide about heme synthesis. It goes counterclockwise, starting from the top left, where you have delta aminolevulinic acid formation. Now, the reason I put this here is because it mentions a number of diseases in blue, collectively referred to as porphyrias, that arise due to abnormalities in any of these enzymes. There are genetic defects where these enzymes are either deficient, mutated, and cannot function properly. So that when heme is being synthesized, there is an accumulation of precursors of heme. These precursors are porphyrins, as you know, and therefore the conditions are referred to as porphyrias. I don't want you to memorize these porphyrias. There are several of them, but in general, these can produce symptoms, and the symptoms tend to be clustered either as neurological symptoms or skin symptoms. And neurological include motor disturbances. They include also psychological and psychiatric presentations like hallucinations and psychosis. The skin, one of the hallmarks of the problems here is sensitivity to light. So they can get ulcerations and alterations in the skin and fragility upon exposure to light. Now, in folklore, and it has been suggested that the myths of the vampires and werewolves were originated in people who had some of these diseases, the porphyrias, particularly of the skin. That's why vampires and werewolves only appear at night, where there's no sunlight, for example. Also, remember what we said earlier, that this first step of synthesis of heme, the formation of aminolevulinic acid, the enzyme there is a rate-limiting enzyme. And we said this enzyme is under the control of heme, such that when you have decreased levels of heme, you have activation of this enzyme. In other words, one way of regulating the activity of the enzyme is to give heme. And if you do that, you decrease this pathway and you decrease the accumulation of the precursors. So currently one of the treatments of porphyrias is to give an analog of heme. Now, if you apply this to the myth of vampires, like Dracula and so on, you would understand why they would like to drink blood, because if you drink blood, you get a lot of heme into the system, and the heme will inhibit the enzyme synthase, and thus they would decrease the the accumulation of the porphyrins, which are the toxic substances that are causing the symptoms of porphyria. But this is conjecture. There's no proof that this is the case, actually. And there is a lot of argument against this, so don't take it as absolute proof. So this is a molecule of heme. Remember, there are these pyrrole rings that are closed. The A, B, C, D are the pyrrole rings. And each of them has substituents or chains on it that include a propionate side chain, a methyl side chain, or a vinyl side chain. The vinyl groups are hydrophobic, whereas the propionate groups are hydrophilic. And then when this binds to globin, it is oriented in such a way that the propionate side chains are directed to the cytosol, to the medium, whereas the vinyl side chains are within the globin molecule. And you notice that iron here is bound and through bonds with the 4 nitrogen atoms of the pyrrole rings. So the hemoglobin chains differ depending on the amino acid composition of those chains. And as I said, the 4 common or relatively common forms in humans, adults, are the alpha, the beta, the gamma, and the delta chains. And the most common hemoglobin, as we said, in adults is hemoglobin A, which is a combination of 2 alpha and 2 beta units. So when hemoglobin formation is deficient, that could be due to iron deficiency, for example, so you cannot synthesize heme, or to one of the deficiencies that we talked about in the enzymes forming heme that cause sideroblastic anemia, or a deficiency in the synthesis of the globins, as you would get in thalassemia. You will see this later. Then the percent of hemoglobin in the cells falls, and hemoglobin constitutes a significant part of the volume of the red blood cell. As a result, the red blood cell volume decreases. And so the number of red blood cells decreases and their volume decreases, and that is what is known as microcytic hypochromic anemia. Microcytic because it is small, hypochromic because its color becomes pale because of the decreased redness due to hemoglobin and to heme. And so this is a little more about hemoglobin. To be more specific, we said there is an alpha 1, alpha beta chains. The alpha 1 and the alpha 2 simply are the same molecule, and the beta 1 and the beta 2 are the same molecule. But we call them alpha 1 and alpha 2 and beta 1 and beta 2 because in hemoglobin, one alpha chain and one beta chain form strong bonds together. They are hydrophobic bonds, but the hydrophobic bonds are so many that it becomes quite a stable interaction. So the alpha 1, beta 1 units, although they are 2 individual units, they actually function more like one unit because they are strongly bound together. Similarly for the alpha 2 and the beta 2. But the alpha 2, beta 2 dimer, if you want, is not as tightly bound to the alpha 1 beta 1 dimer, and so you can think of hemoglobin as 2 dimers interacting with each other instead of 4 different items, although they are actually 4 different units, each one having its own tertiary structure. But when it comes to the quaternary structure, the alpha 1, beta 1 is considered one, and the alpha 2, beta 2 is considered another, and they interact with each other and can move about relative to each other. This slide shows you the synthesis of the different globin chains prenatally and postnatally in humans, and the sites of their synthesis, which are mainly the liver and the bone marrow. In the prenatal period, the liver is the main site for synthesis, and the chains that are predominantly formed are the alpha, as you can see in red, and the gamma chain in yellow. So the hemoglobin of the fetus is made up of the alpha gamma chains rather than the alpha beta, as we said for the adults. Now, there is a beta chain being produced, and it's the minority of the chains. When after birth, the gamma chain synthesis decreases, and the beta chain synthesis increases, so that in the adult, the main chains that are produced are alpha and beta, thus forming hemoglobin A. Fetal hemoglobin becomes very minimal in adult life. Notice that it takes some time for the shutting down all the gamma chain synthesis. So it may persist, hemoglobin F may persist for some time in the newborn. Delta chains continue to be synthesized at a low level, and this may increase later on under certain disease states and can lead to formation of a different kind of hemoglobin called hemoglobin A2. The next slide will explain this. And so the normal hemoglobins in the human adult are HbA, which constitutes almost 98% of total hemoglobin. It's a combination of alpha and beta units. Hemoglobin F, which is found in fetal life and then persists for a while and can be increased in expression in certain thalassemias. It's made of 2 alpha and 2 gamma units. HbA2 is alpha and delta subunits bound together. In addition, the types of the globin chains will also affect the affinity of hemoglobin to oxygen. For example, hemoglobin F has a much higher affinity than hemoglobin A to oxygen. That is not necessarily a very good thing, because hemoglobin has to, yes, it has to bind, it has to have affinity, but it also has to release it. Otherwise, it will lose its function. Hemoglobin has to deliver oxygen to the tissues. So there must be it must bind it under certain conditions and release it under other conditions. Now, there are a number of abnormal hemoglobins that can be produced due to genetic mutations. They are called hemoglobin C, D, E, H, etc. And hemoglobin S, which is known as sickle hemoglobin, which you encounter in sickle cell disease. It is the most important one clinically, and it's a cause of sickle cell anemia, a genetic problem where there is a mutation where valine is substituted for glutamic acid at one point in the beta chains of hemoglobin. So this is a problem with the beta chain. So, and when this happens, what happens is that when there is low oxygen tension, some kind of hypoxia or decreased ischemia to tissues, what happens to the hemoglobin is that it forms crystals. It aggregates inside the cells, and these can be quite long. This causes deformation in the red blood cell, what we call sickling. It becomes like a sickle, the thing you use in farming, a crescent, yeah. And these, when this happens, the cells are deformed and they can block capillaries and cause ischemic changes, decreased perfusion of tissues, and the spikes can rupture the cell membranes, and this leads to anemia. So you get sickle cell anemia. So with this introduction, let's now move on to see how hemoglobin and oxygen interact. So as we said, the most important feature in the hemoglobin molecule that makes it a good carrier of oxygen is that it has to combine with it, but it has to bind it loosely and reversibly. Because yes, you want to carry the oxygen, you have to bind it, but then at some point you have to release it. So it has to be a reversible and not too tight binding. That's why we said earlier when we talked about hemoglobin F having much higher affinity than hemoglobin A, this may not be a very good thing to serve as a carrier of oxygen and a releaser of oxygen to the tissues. Now, every iron atom in the heme molecule can bind one molecule of oxygen. And so every hemoglobin molecule can carry 4 molecules of oxygen, whereas every myoglobin molecule can carry only 1 molecule of oxygen, because myoglobin is only one single chain with one heme group in it. And as we said, the primary function of hemoglobin is to bind the oxygen in the lungs as much as possible, and then carry this to the tissues and then release it in the tissues. Now, the binding of oxygen to hemoglobin is unique or very special. And this is a kind of binding or kinetics of binding that you have not been introduced to before, I think. We have talked about binding of compounds and molecules to receptors and to transporters and to enzymes and so on. But most of the discussion has been where one molecule binds to one receptor at one site. And this is described usually by a hyperbolic curve, where as you increase the binding, or as you increase the concentration, the binding increases until it reaches saturation. Well, this also happens with hemoglobin, except that when one molecule of oxygen binds to one heme group, this binding itself is going to increase the affinity of the other heme groups in the hemoglobin molecule to molecules of oxygen. So the other heme groups now have an increased affinity, so more avid binding to oxygen. And the same happens in the reverse. When you dissociate oxygen from a heme group in the tissues, this dissociation is going to decrease the affinity of the other heme molecules to oxygen, and therefore there is a further release of oxygen in the tissues. This is called cooperative binding, and it is described by a special curve. This is not the normal hyperbolic curve that we see with drug receptor interactions. It is a sigmoidal curve. So this change in the affinity of hemoglobin to oxygen as oxygen binds to the heme groups is related to conformational changes that happen when an oxygen binds to its heme group. So in the deoxygenated state, when there is no binding of oxygen to hemoglobin, we say that hemoglobin is in a state. We call it a T state or a tense or a taut state. The two alpha beta dimers are linked. We said the two alpha beta dimers, alpha 1, beta 1, are linked together by hydrophobic groups, and the alpha 2 and beta 2 are linked together by hydrophobic groups. But the two dimers, alpha 1, beta 1, and alpha 2, beta 2, also interact through hydrogen and ionic bonds. Now, when heme binds the oxygen, when the first molecule binds to heme, there is a conformational change that happens, leading to the breakage of the chains or the bonds that bind the two dimers, the alpha 1 beta 1 to the alpha 2 beta 2. And there is a rotation that happens in these molecules to each other, around 15 degrees rotation. This leads to the R state, and the R state, or the relaxed state of hemoglobin, is associated with increased affinity of hemoglobin to oxygen. And this explains the cooperative nature of the binding. Now, this is a little more elaboration. Now, on the left side is heme without the binding of oxygen. Now the heme is the iron is the green molecule, which is bound to the series of black molecules there on the plane. This is the porphyrin, and below it is a histidine. This is a histidine that belongs to the globin chain. So if you remember, looking at the figure in the top, the protoporphyrin, the heme molecule, consists of the protoporphyrin and the iron in the middle. And the iron is bound to the 4 nitrogen groups in the plane, almost in the plane of the molecule. Now, there are 2 more bonds that iron can form. One of them is a bond with the histidine group that belongs to the globin chain. And there's a 5th site, or a 6th site at the top, which is free. That's where oxygen binds. On the right side, oxygen in the red molecules binds to iron. And notice what happens when oxygen binds to iron. The iron moves in the direction of oxygen so that it becomes much more in the plane of the porphyrin molecule. On the left side, it was below the plane. Here, it's in the plane. And when it moves with it the histidine group of the globin chain. And when this happens, this tugs on the whole globin chain and results in a change in the conformation and a breakage of the bonds between the 2 dimers of hemoglobin. And this results in the conformational and the rotational change that leads to increased affinity of the other heme groups to the oxygen molecule. So for oxygen binding to happen, you should even know that iron must be in the ferrous state. If the iron is in the ferric state, it cannot bind. It cannot bind to oxygen. And this is in contrast to the heme that is found in enzymes and other enzymes like cytochromes and oxidases, where the iron shifts between ferrous and ferric, and that's important for its function. That's a different discussion. But for the function of hemoglobin, the iron has to be in the ferrous state. If the iron becomes ferric, then you lose the ability to bind oxygen. There are certain situations where iron can become ferric, and we'll talk a little more about them in just a second. And remember also that there are other compounds other than oxygen that can bind to heme. One of the notorious compounds is carbon monoxide, CO, but also nitric oxide and hydrogen sulfide can also bind. And these can be toxic compounds if inhaled. Carbon monoxide specifically can be very toxic, as we will discuss later on, even at very small concentration. And they bind to heme not only in hemoglobin, but also in the other enzymes that are essential, some of the cytochromes important in the oxidative phosphorylation pathway, and they can be toxic there as well. Now, as I said, iron has to be in the ferrous. If you oxidize it to ferric, and this can happen under oxidative stress or by some drugs can do that, you result in the formation of what is known as methemoglobin. Methemoglobin is hemoglobin where the iron is in the ferric state. And this methemoglobin cannot bind oxygen. So this is an unwelcome situation. By the way, methemoglobin has a brown color, and that's what accounts for the color of old meat or which has been exposed to air, or to dried blood, where the blood becomes, instead of red, becomes brown. That's due to the methemoglobinemia, methemoglobin formation. Methemoglobinemia means increased levels of methemoglobin in the blood. Normally, this does happen sometimes at low levels, but the RBCs contain an enzyme called methemoglobin reductase that can convert the methemoglobin back into the normal hemoglobin by reducing the ferric to ferrous. Now, let's go back to the transport oxygen and role of hemoglobin in oxygen transport. So as we said, in the lungs, oxygen is being, you're breathing air, oxygen is coming in, and it's diffusing from the lungs, the alveoli, into the pulmonary blood. Here it binds to oxygen, to hemoglobin, and gets transported. It can also dissolve in the blood, but the amount that dissolves in the blood is much less than that is carried by hemoglobin. It accounts only for 3% of the total transported oxygen, whereas oxygen bound to hemoglobin accounts for 97% of what is carried by the blood as a whole. So that's why hemoglobin is so important, because otherwise you would not be able to deliver oxygen appropriately to the tissues that need it. So when PO2 is high, PO2 is the partial pressure of oxygen, which reflects its concentration actually also in the blood. When the PO2 is high, as you would get, for example, in the lungs, in the pulmonary capillaries, oxygen binds with hemoglobin. However, when the PO2 is low, as you would find in the tissues where oxygen is being consumed, then hemoglobin can no longer bind oxygen very tightly, and oxygen is released. This is a reversible, as we said, interaction. It has to be reversible, such that when you decrease the tension of oxygen or the partial pressure of oxygen, oxygen gets released from hemoglobin. Just as you would, the same relationship that we discussed with protein binding of drugs, for example. So, and this relationship is reflected in what is known as the oxygen hemoglobin binding curve, or more commonly it's referred to the oxygen hemoglobin dissociation curve. It's both the binding and the dissociation curve. And this is it, depending on whether you're going from the left to the right, which means you're binding oxygen, or right to left, which means you're dissociating. And what is this curve? This is relating the partial pressure of oxygen in the blood, PO2, to the degree of hemoglobin saturation with oxygen. So as the partial pressure of hemoglobin increases, the hemoglobin saturation increases, and it reaches eventually a maximum at very high partial pressures of oxygen. And this is the sigmoidal curve we mentioned earlier. So in the lungs, where the partial pressure of oxygen is very high, oxygen is almost 97% saturated. The partial pressure of oxygen is around 95 or 97 in the blood, in the lungs, in the blood perfusing the lungs. And so as you can see, the saturation of oxygen is around 95, 97%. In the tissues, the partial pressure is 40 under normal conditions. And so the saturation of oxygen goes down to 75 or something like that. So as you can see, there is a decrease in oxygen saturation. Oxygen is being released from hemoglobin as it goes from the lungs to the tissues. And what you release is almost 22-25% decrease in the saturation. This is what happens under normal circumstances when you are mostly at rest. So let's do some calculations here. So the blood of a normal person contains approximately 15 grams of hemoglobin per 100 milliliters of blood. And we know that 1 gram of hemoglobin can bind to a maximum with 1.34 milliliters of oxygen. So if the hemoglobin is 100% saturated, how much oxygen is it carrying? So it means that every 100 milliliters of blood, which has 15 grams, will carry a maximum of 20 milliliters of oxygen. If 1 gram carries 1.34, 15 will carry 20 milliliters of oxygen if it is 100% saturated. So if you go back to the oxygen dissociation curve, we said the oxygen saturation decreases from around 97% in the lungs to around 70-75% in the venous blood. So if you calculate how much oxygen is being released, it turns out to be around 5 milliliters of oxygen per 100 milliliters of blood. That is what is needed by your tissues under normal resting conditions. And that is achieved by hemoglobin just releasing around 22% of its content. So it retains a lot of oxygen in it. And this is because the saturation curve that we saw earlier, the oxygen dissociation curve, is a sigmoidal curve where at high oxygen levels, at high PO2s, the curve decreases in a shallow, with a shallow slope, right? So for a long, for a wide range of PO2s, the saturation of oxygen changes slightly. So with this in mind, let's just reiterate what we said earlier, that almost 97% of oxygen is carried by hemoglobin and only 3% by dissolved in the blood. So at a PO2 of 95 millimeters mercury, actually only 0.29 milliliters of oxygen can be dissolved in every 100 milliliters of plasma or water of the blood, right? At the PO2 of 40, this goes down to 0.12. So that's what is found in the tissues. So from the lungs to the tissues, how much are you removing? You're removing only 0.17 milliliters of oxygen for each 100 milliliters of blood. Contrast this with the 5 milliliters of oxygen that are released by hemoglobin when you go from the lungs to the tissues. So 5 versus 0.17, that's almost 97% versus 3% of the total needs of the tissues, which are subserved by hemoglobin carrying oxygen and cannot be achieved if oxygen were only to be dissolved in the water of the blood. Okay, so that was what's happening under resting conditions. Under resting conditions, with the normal venous blood, you only need to go from 100 or 97 PO2 to 40 PO2 so that you can deliver the needs of the body in terms of oxygen. What happens during exercise? Well, during exercise, you have to utilize more oxygen, and therefore there's increased utilization of oxygen, and the PO2 falls to around 17 millimeters mercury. Now, what happens to the saturation of oxygen? Because of this curve, which now becomes a much steeper curve, a much smaller decrease in PO2 is going to cause a much greater reduction in the saturation of hemoglobin. So you lose a huge amount of oxygen that's found in the hemoglobin, going down to a saturation of much less than you achieved earlier. So during exercise, therefore, you increase the delivery of oxygen to the exercising muscles, and you do this by two ways. By decreasing the PO2 further, which means you decrease the saturation of oxygen and you release the oxygen from the hemoglobin along the steep curve. But also remember that during exercise, there is a marked increase in cardiac output. So there is increased delivery of blood. So these 100 milliliters are being delivered at a much faster rate. And therefore, you're increasing the delivery of oxygen as well to the tissues. And as a result, as we say here, let's analyze it the way we did for the resting situation. At a PO2 of 15 millimeters mercury, what remains is only 4.4 milliliters of hemoglobin instead of 19.4 that you started out with in the lungs. And so you have delivered 15 milliliters of oxygen for every 100 milliliter of blood delivered to the tissues. This is 3 times what you normally do under resting conditions. And remember that the cardiac output can increase up to 6 times sometimes. And so basically you're increasing the delivery of oxygen up to 20 times, 18 or 20 times to the tissues when you do exercise. So hemoglobin, therefore, if you look at the curve, it serves really as a buffer, not only for acid base, but also for tissue oxygen. So if alveolar PO2 is decreased to 60 millimeters mercury, not 100, and if there is a situation where there is a decreased oxygen supply, the arterial hemoglobin is still 89% saturated if you look at the oxygen dissociation curve. At P60, you have 89% saturated, only around 8% decrease from the normal. So even at this low PO2, even under these conditions of decreased oxygen, the tissues can still extract the 5 milliliters of oxygen per 100 milliliters of blood simply by decreasing the PO2 to 35 millimeters mercury instead of 40 millimeters mercury. So the PO2 doesn't have to change that much in order to maintain a physiological situation. And in fact, if the alveolar PO2 changes between 60 to 500, you find hardly any change in the PO2 from normal. And this is what we call the oxygen buffering function of the hemoglobin. This is thanks to its sigmoidal curve. Okay, so let's discuss a bit hemoglobin versus myoglobin. As we said, myoglobin differs from hemoglobin in that it has only one heme, and therefore its binding is the normal binding that we studied earlier on, which is this hyperbolic curve on the left, the black curve. Hemoglobin is the sigmoidal curve on the right. Notice that the P50, the PO2 at which you get half saturation of the hemoglobin, is much lower for myoglobin than for hemoglobin. And that for myoglobin, no matter how much you decrease the PO2, going from 90, say, to 20, you still do not release much oxygen to the tissues. It is only when you get to very low levels of PO2 that you get this precipitous drop in the saturation of myoglobin. And that's what we meant when we said that myoglobin actually is not a good carrier and deliverer to tissues, but it's a good storage for oxygen, because when the tissues are so poorly supplied with oxygen and the PO2 is very low, that's when myoglobin steps in and delivers the oxygen to the muscles so that they can use them and function normally. So this slide simply reiterates what we have just discussed, being about the higher affinity and the hyperbolic nature of myoglobin, which makes it a poor oxygen transporter, but a good store for oxygen. Now, there are many factors that can shift the hemoglobin oxygen curve, dissociation curve. Some shifted to the right, downwards, and some shifted to the left and upwards. Things that shifted downwards and right include acidosis, that is increase in hydrogen ions, increased CO2 levels, increased temperature, and increased concentrations of a specific metabolite called BPG. We'll mention this in a while. How does this work? The increase in hydrogen and CO2 causing a decreased affinity to oxygen is referred to as the Bohr effect. That is the shift of the O2 curve to the right in response to these changes. So what happens? In the tissues which are functioning, CO2 is being produced. When CO2 is produced, it diffuses to the blood, and this increases the pCO2 in the blood, the partial pressure of carbon dioxide. As a result, there is formation by combination of CO2 and water of carbonic acid, H2CO3, which then dissociates to give H plus and HCO3 minus, bicarbonate and hydrogen ion. The rise in H plus shifts the oxygen dissociation curve to the right and downwards, so that more oxygen is released at any PO2 level. So when you have acidosis, when you have increased CO2 production, which means the tissue is active and is requiring a lot of oxygen, what's happening is that you're shifting the curve downwards so that you are releasing more oxygen to the tissues at that pH. In the lungs, the opposite happens because in the lungs, CO2 is going to move out to the alveoli from the blood to the alveoli by respiration and diffusion. This decreases the pCO2 in the blood, and as a result, it decreases the hydrogen ion, which shifts the curves back to the left and upwards so that more oxygen becomes bound to hemoglobin at any PO2. And that is the function of hemoglobin. In the lungs, it has to bind it, and in the tissues, it has to release it. So let us elaborate a little more about CO2 and how it is handled by the hemoglobin and the blood. So this slide shows you that CO2 is being produced by the cell in the tissues, and the CO2 thus produced diffuses into the blood. Now in the blood it has three fates. One of them, depicted by the arrow going downwards, is dissolving in the water of the blood. But this accounts for very little of the transport of CO2, around 7%, as you can see in the table on the right. The other, what is left of CO2 can have two fates. One of them is binding of CO2 to hemoglobin itself to form what is known as carbaminohemoglobins, CO2HB, and that is approximately 23% of the total. The majority of CO2 is going to form carbonic acid, and it does this by combining with water under the influence of an enzyme that is found in the RBCs called carbonic anhydrase. The formation of carbonic acid can happen without the enzyme, but the presence of the enzyme makes it a much faster, thousands of times faster process. Carbonic acid then dissociates into H plus and bicarbonate HCO3 minus. HCO3 minus goes out to the blood, and the hydrogen ion binds to hemoglobin because hemoglobin in the deoxygenated state becomes more basic. In the oxygenated state, it becomes more acidic. So in the deoxygenated state, it accepts hydrogen and you get binding of hydrogen to hemoglobin. So as you can see, this is the role of hemoglobin and the RBC as a buffer, an acid-base buffer in the blood. So now we can talk about another interaction, the Haldane effect, which is sort of opposite to the Bohr effect. The Haldane effect tells you how binding of oxygen affects the binding of CO2 to hemoglobin. And basically this is it. When the blood reaches the lungs, oxygen is in high amount there, and it will bind to hemoglobin. This binding to hemoglobin, as we said earlier, is going to make it a stronger acid. So as a stronger acid, it will release the hydrogen that was bound in the tissues, and the hydrogen that was released then will bind to bicarbonate in the blood to form carbonic acid, which then dissociates into carbon dioxide and water, and the carbon dioxide gets washed out through the lungs. At the same time, the more acidic hemoglobin becomes less bound to carbon dioxide. It decreases the affinity to carbon dioxide. So carbaminohemoglobin will dissociate and will release the CO2. So this way you have transported carbon dioxide from the tissues to the lungs so that you can exhale it in the lung and get rid of it through both hemoglobin and its function as an acid-base buffer through the bicarbonate. Now, BPG, the other thing we mentioned that affects the curve, is bisphosphoglycerate, also called DPG or diphosphoglycerate. This is an intermediate in the metabolic pathways of the cell. It's a side arm of the glycolytic pathway where 1,3-DPG gets converted to 2,3-DPG. Um, well, this usually happens under hypoxic conditions. Again, so when you have hypoxia, you have increase in 2,3-DPG or a decrease in PO2, let us say. The decrease in PO2 increases the concentrations of 2,3-DPG through this product side arm of glycolysis. The 2,3-DPG then shifts through interacting with hemoglobin, shifts its dissociation curve to the right, decreasing its affinity to oxygen, and then releasing oxygen to the tissues. Again, this is another important adaptation to hypoxia and to increase metabolic activity of the cell. And you see it when you have decreased tissue perfusion, blood flow. You get increased concentrations of 2,3-DPG. Now, interestingly, we mentioned earlier that oxygen is not the only one that binds to heme, only gas that binds to heme at the iron site. Carbon monoxide can bind at exactly the same site as oxygen. The problem with carbon monoxide is that it binds with a much, much higher affinity, around 250 times higher affinity. Notice the curve and the PCO50, which is around 0.15 or 0.14 millimeters mercury. Contrast this with what you see with the oxygen dissociation curve. The PO250 is around 20 or 25 or 30. So it's around 250 times higher in affinity, which means that at a PCO of 0.4 millimeters mercury only, this is 1 over 250 of the alveolar PO2, the carbon monoxide can compete equally with oxygen for hemoglobin. So if you raise this PCO just slightly at 0.6 millimeters mercury, this can be a lethal level in the air. That's around 1 part per 1,000 in the air. And that's why carbon monoxide is such a lethal gas. It is lethal because, particularly with carbon monoxide, it has no smell, and the patients do not show the signs of hypoxemia. The PO2 remains normal in the blood, but the oxygen bound to hemoglobin is not there. CO is binding instead of it. The blood appears red. There is no cyanosis, no blue discoloration of the lips and the tips of the fingers that you normally get when the PO2 decreases, and the blood is bright red. So again, because the PO2 is normal and PO2 is one of the signals to stimulate breathing. So if there's a decrease in PO2, you get a stimulation of breathing. Here, the PO2 is normal, so the breathing is normal. So you're not hyperventilating, you're not trying to compensate for the loss of oxygen. So this is very dangerous. The patient gradually sinks into unconsciousness without realizing that there is anything wrong. And that's because of the decreased oxygen supply to the brain, of course. And if unchecked, they can die. So if I want to ask you a question, how would you treat a patient poisoned with carbon monoxide? What would be the ideal way to do it? One of the answers, the answer here is you treat with 100% oxygen. You have the patient breathe 100% oxygen. And that is basically just applying the principle of competition. You're raising the concentration of oxygen, the normal ligand for hemoglobin, to displace the carbon monoxide from hemoglobin. And in some cases, they add a little of carbon dioxide, 5% carbon dioxide, because increased carbon dioxide can lead to stimulation of respiration, just as decreased oxygen partial pressure can stimulate respiration. So CO2 breathing activates the respiratory centers, so you breathe more rapidly, and so you wash out the carbon monoxide more rapidly from the blood, almost 10 times faster. But O2 also breathing 100% oxygen for a prolonged time itself can be quite toxic to some tissues due to oxidative damage. So this is practiced for a short period of time. The last chapter in this lecture will talk about heme metabolism. So what happens when RBCs die, when hemoglobin is broken down, and what are the clinical relevance? What's the clinical relevance of it? So as you know, red blood cells normally live for around 120 days. And then when they become senescent, they are taken up by macrophages in the reticuloendothelial system of our body, mainly the spleen and the liver, where they are endocytosed and degraded. And as a result, hemoglobin is released and is broken down into its constituents, which are the globin chains and heme. The globin chains are then reduced into their amino acids, which join the amino acid pool of the body. And heme is then broken down into iron, which again joins the pool of iron in the body, which we will discuss in the next lecture on iron metabolism, and porphyrins, which are transformed into the final product, which is bilirubin. And bilirubin is termed here unconjugated. It refers to the bilirubin is released into the plasma and gets bound to albumin. So all bilirubin in the plasma is bound to albumin. It cannot exist in the free form as such. But when we talk about bilirubin, we talk about unconjugated and conjugated bilirubin. The unconjugated bilirubin is also referred to as free bilirubin, but this doesn't mean totally free. It means the bilirubin that is bound to albumin. We call it free to distinguish it from the conjugated form, which is conjugated to a glucuronide moiety in the liver. Now, this represents the metabolism of heme. Heme is converted first to biliverdin through a heme oxygenase, and that compound is then converted to bilirubin through the biliverdin reductase. The bilirubin is then transported to the liver combined to albumin. In the liver, it is acted upon by a glucuronide transferase to become conjugated bilirubin. And this is what happens to bilirubin that is conjugated in the liver after its delivery from the periphery. Conjugated bilirubin is secreted into the bile. The bile delivers it to the intestines. And in the intestines, this bilirubin can be converted to a product called urobilinogen. Urobilinogen is then either excreted as such, or it is converted to stercobilin. Stercobilin is a pigment that gives the feces its color, its normal color of brown, dark brown. Urobilinogen can also be reabsorbed through the enterohepatic circulation back into the blood, where it can be taken up by the liver and secreted again, or in the blood it can be transported and can be eliminated in the kidney. So under normal circumstances, there might be a small amount of urobilinogen in the kidney, in the urine. Now, remember this pathway because it will be relevant when we discuss jaundice. Jaundice refers to this yellow tint that happens when levels of bilirubin are increased in the blood and in the extracellular fluid and in the tissues. So this is a yellowness of the skin as well. If you look inside, there might be yellowness of the organs as well. As we said, the usual cause an increase in bilirubin, and this bilirubin can be either the conjugated or the free bilirubin. Both of them can induce this. Normally, the levels of bilirubin in the plasma are approximately 0.5 milligrams per deciliter, and most of this is in the free form, the unconjugated form, bound to albumin. When the levels increase, and particularly above 1.5 milligrams per deciliter, that's when you start seeing the actual signs of jaundice, the discoloration of the skin and of the particularly of the sclera, the white of the eye. And this is a typical patient presenting with jaundice. You notice the yellow discoloration of the skin and also called icterus. This yellow discoloration, icterus. This patient happened to have a cancer of the pancreas that was obstructing the common bile duct and resulted in obstruction to bile flow. And as a result, the bile pressure increased and the bilirubin flowed back into the plasma. And as a result, they get jaundice. So when patients present with jaundice, these patients conceivably can have one of two basic problems or causes. There is either too much production of bilirubin at a rate that cannot be handled by the elimination processes, or there is a problem with the elimination of bilirubin from the body. So as far as this current course goes, the blood course, most of what you'll see is the first, which is due to hemolysis. Hemolytic anemias may be associated with jaundice. That's because in these cases you are getting increased rate of destruction of RBCs and therefore greater metabolism of heme. And in these cases, obviously, the bilirubin will be predominantly in the free form, not the conjugated form. The other general etiology is hepatic or post-hepatic and involves obstruction or decreased elimination of conjugated bilirubin. And this results in overflow of this conjugated bilirubin. It cannot be released to the bile and to the intestines. It overflows into the plasma and raises the levels of conjugated bilirubin. So this you'll learn more about in the GI course. And the lab tests, the clinical tests that we have for measurement of bilirubin are able to measure both the unconjugated and the conjugated forms of bilirubin. And the levels will tell you or will point you towards an etiology for the jaundice that you are investigating. In hemolytic jaundice, due to hemolytic processes, you expect to see more of the free form or the albumin-bound form, whereas in obstructive jaundice, it is mainly in the conjugated form. And you will learn more about the use of bilirubin in the GI course because it's not always black and white. If there is complete obstruction, remember what we talked about, what happens in the intestine. A patient who has complete obstruction means there's no bilirubin reaching the intestines, and therefore there's no production of urobilinogen or of stercobilin. And stercobilin is the pigment that gives feces its color. So a person who presents with jaundice and what are known as clay-colored stools is most likely having an obstructive lesion. And this person is not expected to have much urobilinogen in his urine because urobilinogen is not being produced in the intestine and is not being reabsorbed and transported to the kidney for excretion. So with this, we end the lecture on hemoglobin and myoglobin and heme. And next will be our discussion of iron metabolism and iron deficiency anemia.